as a cytotoxic reaction. Type 2 hypersensitivity is also called as a cytolytic or cytotoxic reaction because it results in the destruction of host cells either by lysis or toxic mediators. Type 2 hypersensitivity is caused by antibodies binding to cells or tissues antigen. The antibodies are of the IgM or IgG classes and cause cell destruction by Fc dependent mechanism, either directly by recruiting the complement via the classical pathway. And classical example of type 2 hypersensitivity reaction are the responses exhibited by a person who received a transfusion with the blood from donor with different blood group. And second example is the erythroblastophytalis. There are the two different antibody mediated mechanisms are involved in this cytotoxic reaction. In complement mediated hypersensitivity reaction, antibodies react with the cell membrane components. Okay, and leading to the complement fixation. Okay, see here antibodies are react with the cell membrane component, and when they react with the cell membrane components, there is an activation of complement protein which activate the classical pathway of complement. Okay, and there is a formation of complement cascade and leads to the either to lysis of the cell or opsonization. Okay, this is called as a complement mediated type 2 hypersensitivity response. Okay, second one is the antibody dependent cell mediated cytotoxicity. So in this second subtype of the type 2 hypersensitivity, they use the Fc receptor expressed on the many cell. Okay, see here, this is the here for example here is the natural killer cell which possesses the Fc receptor. Okay, these Fc receptors are also present on the neutrophils, eosinophils, macrophages, etc. As means of binding, bringing these cells in contact with the antibody-coated target cell. Lysis of these target cells required contact, but does not involve phagocytosis or complement fixation, which observed in the complement-mediated hypersensitivity. Instead, ADCC lysis of the target cell is analogous to that of cytotoxic T cells and involves the release of cytoplasmic granules containing protein, for example porphyrin and granozymes, that activates events leading to the apoptosis. Okay, so this mechanism is called as an ADCC reaction, involve IgG and IgG Fc receptor. Okay, this is all about the type 2 hypersensitivity reaction. Next is the type 3 hypersensitivity reaction. So type 3 hypersensitivity and type 2 hypersensitivity, there is a slightly difference in both. First one is the types of antibodies are involved. In type 2 hypersensitivity, two antibodies or two immunoglobulins are involved, which include IgG and IgA. And second is the type of antigen. In type 2 hypersensitivity, there are the two types of antigens may involve. First one is the cell which shows the autoimmune reaction, and second one that is the maybe intrinsic in nature, and next one is the extrinsic antigens, okay, which are recognized as a non-cell. Okay, in type 3 hypersensitivity reaction, the types of antigen which is involved is mostly the insoluble antigen, and the types of immunoglobulin which is involved is mostly the immunoglobulin G. Okay, so what is the definition of type know it is the antigen antibody or antigen antibody complex is also called as a immunocomplex. So type 3 hypersensitivity is the immunocomplex or antigen antibody mediated reaction. It is a mechanism in which small antigen antibody complex produced from the normal immune response to foreign substance deposit and accumulate in the different tissue, triggering a maladaptive immune response. Okay, so what is the exact mechanism? So, for example, when IgG antibodies are produced from a normal humoral response to foreign or self antigen. Okay, for example, SLE disease. Okay, that is the example, one of the example of autoimmune disease. At that time, IgG binds this antigen, forming an antigen antibody complex, and the body response can vary depending upon the size of antigen antibody complex. So, body show the two types of response. When antigen antibody complex is large, okay, that time it is easily cleared by the reticuloendothelial system in the spleen, and no allergic manifestations are observed. But when antigen antibody complex is small, okay, or when small immunocomplex go unnoticed by the reticuloendothelial system and it remain in circulation because its size is small. At that time, these immunocomplex deposit and accumulate in body tissue. So, because of that, what happened? IgG immunocomplex degranulated in mast cells and histamine is released. Or when IgG in immunocomplex activate the complement cascade. And because of these both type of reaction, there is the inflammation of tissue is noticed. Okay, and because of that, there are different types of different types of hypersensitivity develop. For example, if self antigens is the cause, okay, at that time it is called as a systematic lupus erythematosus disease. That is also called as a SLE. Okay, and if antigen is the bacterial antigen, okay, at that time the hypersensitivity develops. It may be the reactive arthritis, post-infection glomerulonephritis, and serum sickness. Okay, this is all about the type 3 hypersensitivity reaction. Once again, I repeat in short, the type 3 hypersensitivity, it is the immunocomplex mediated hypersensitivity. It is the mechanism in which various tissue sites where they activate the complement and subsequently cause the tissue cell lysis. Normally, these complexes are phagocyted effectively by the fixed monocytes and macrophages of the monocyte macrophages system. In the presence of excess amount of soluble antigen, the antigen antibody complex may not be efficiently removed. There is the accumulation can lead to the hypersensitivity reaction from the complement activation. So this is the type 3 hypersensitivity. In next video, we will discuss about the type 4 hypersensitivity and overview of the hypersensitivity reaction.